perhaps a cumulative, shall we say, allostatic load that will make that brain more vulnerable to damage later on. So it's not without its negative consequences. Now, just to say a little bit, because it's very important that you understand that this has there is information for the human brain that these studies in animals predict something that goes on in the human brain. For example, in Cushing's disease, there is hippocampal shrinkage. In type 2 diabetes, the hippocampus gets smaller. In major depression, the hippocampus gets smaller. In post-traumatic stress disorder, there's evidence that a smaller hippocampus may be a risk factor for PTSD. It may also be a consequence of the chronic stress associated with the aftermath of PTSD. And as an illustration of the diabetes end of things, this is work by Antonio Convite's group at New York University Medical Center, showing that as people who have higher levels of glycosylated hemoglobin, in other words, poor glucose control, the higher the level of glycosylated hemoglobin, the smaller is the hippocampal volume, there is memory impairment, and also there appears to be increased risk for later developing dementia. And what you also need to know about the hippocampus is that it's a target for not only for insulin, but also for ghrelin, a gut hormone, for leptin, a fat tissue hormone, and normally these are able to get into the hippocampus and have positive effects on mood and memory, induce structural changes, protect or promote efficient glucose uptake, and in the case of IGF-1, there's evidence that it mediates exercise-induced neurogenesis. In other words, when you go out and are physically active, there's IGF that gets into the hippocampus and helps to enhance the neurogenesis that can keep your hippocampus, your dentate gyrus, healthy and happy. But then there are other conditions that are not disease per se. There's evidence that chronic stress over many years causes a smaller hippocampus. Chronic inflammation is associated with a smaller hippocampus. Jet lag with a smaller hippocampus. Lack of exercise with a smaller hippocampus. And low self-esteem also with a smaller hippocampus. And to show you one study on jet lag, chronic jet lag, a study on airline personnel showed that air crews that have less than a 5-day period to recover between international flights actually show a smaller hippocampus, a smaller temporal lobe in which the hippocampus is located. They have memory impairments, they have elevated levels of cortisol, compared to a group of the same air crews that have 14 days or longer to recover between flights. So this is very important for work rules for people to keep their brains healthy. And finally, the news is not all bad, because there's a study in Germany showing that medical students who are studying for an exam over a 3-month period, intensive studying, they show increased volume of their parietal cortex and their posterior hippocampus, which is this area that's particularly important for learning and memory of events and probably facts. And they take an exam at the 3-month period, and then you see the parietal cortex over the next 3 months doesn't get any smaller, doesn't continue to increase, but the hippocampus keeps getting somewhat larger. Now, this could be due to increased neurogenesis, increased growth of dendrites and synapses, more glial support cells, and a more efficient blood supply. We don't know yet, but probably all of the above, based upon things we know from other animal experiments, putting animals in enriched learning environments. So let's talk then about another brain area, the amygdala, which communicates with the hippocampus, which also communicates with the prefrontal cortex, and is also subject to the effects of chronic stress. So for example, the same chronic stress that causes neurons in the hippocampus to shrink causes neurons in the basolateral amygdala to grow and develop more synaptic connections. This is work of a colleague in India, Sumantra Chatterjee, and was first published in 2002, and there have been a lot of work since showing this, amplifying on this. Even a single traumatic stress, taking a rat, putting it into an immobilization bag to get a blood sample, causes over 10 days increased anxiety and induces new synaptic connections on these dendrites. Doesn't increase the length of dendrites, it takes repeated stress to do that, but in both cases the effect of this traumatic stress exposure is increased anxiety. Now the human brain, the amygdala responds, for example, to the sight of what might be a snake with a fear response, freezing and then backing away. The amygdala is also subject to reacting to angry and fearful faces that a person may be shown on a screen while lying in an MRI machine. And as you remember, the amygdala is important for fear, anxiety, and aggression, for autonomic activation, for fear conditioning, and also is overactive in chronic anxiety. And so facial expressions are used to activate not only the amygdala activity, but also to activate heart rate and blood pressure and autonomic activity, skin responses. And what happens, what has been found among many things, is that people who are vulnerable for atherosclerosis, they have beginning signs of atherosclerosis and early stages of cardiovascular disease, actually are more responsive to these angry and fearful faces than people who do not show these tendencies. So the idea is that perhaps as people are exposed in their daily lives to seeing people with facial expressions and emotions, those people are particularly susceptible, and they show the development of an allostatic load each time with an increased heart rate and blood pressure response, which can ultimately cause some damage to the cardiovascular system. So what we have then is a situation where chronic stress causes neurons in the hippocampus to shrink, neurons in the basolateral amygdala to grow. The animal becomes more fearful, less able to remember things. There's also an area of the brain called the orbital frontal cortex, which is linked to the amygdala, which also shows this growth response. And the medial prefrontal cortex also responds like the hippocampus, and those neurons shrink. And I don't have time to summarize in detail what's going on, but we know that this shrinkage of dendrites, which leads to impairment of cognitive flexibility, is reversible when you stop the stress in young adult rodents. We know in a study on medical students studying for the board exam, done just next door at Cornell Medical School, that there is also reversible plasticity in a circuit that involves their prefrontal cortex after chronic stress that suggests that the same thing is happening in the young human brain. But there's also, in the rodent models, a loss of the resilience with aging. The middle-aged and older rat shows a shrinkage of dendrites, but it does not recover very fast, if at all. We also know that circadian disruption, as in jet lag or shift work, also causes these medial prefrontal cortex dendrites to shrink, and it also impairs cognitive flexibility, the ability to shift when something different it challenges the brain, the individual. And finally, in our rodent models, we know that females do not respond the same way to males. All of the studies that I've described so far are done on male rodents, and we have a lot to learn about how the female brain responds. Talk briefly about interventions, because top-down interventions are very important along with pharmaceutical agents For example, regular physical activity has been shown, and this is not becoming a marathon runner, but simply walking 60 minutes a day, say for 5 days out of 7 a week, enhances executive function, improves blood flow to the prefrontal cortex, enhances neurogenesis in the hippocampus, and in fact a recent study has shown that getting sedentary adults in their 60s to walk 60 minutes a day over a year will actually increase the volume of the hippocampus and improve their memory. We also know from programs like the Experience Corps, getting elderly adults to volunteer to become, for example, teachers' aides in an elementary school has the beneficial effect to their brain function, improves both their physical and mental health, and in fact produces many of the changes that are described here to their prefrontal cortex, to their ability to make decisions, and also shows some protection from dementia, that development of dementia having meaning and purpose in life. And then there are studies using cognitive behavioral therapy, getting people to reduce their stress by mindfulness and other methods, showing that this also can change the volume as well as the function of key brain areas like the amygdala. One of the important themes of this talk is to go back to early life events and talk about how this can have lifelong influences, not only by increasing allostatic load, cumulative damage, but also by biological embedding, things that happen very early in life that can have later adverse consequences. We know that undernutrition of the fetus, as in the Dutch hunger winter, overnutrition, as many cases now where people are, pregnant women have too many calories, can also have effects on the vulnerability to things like diabetes and cardiovascular disease. We know that maternal stress has adverse effects. We know that chaos in the home also has adverse effects, that abuse and neglect have adverse effects that last for many years. Low socioeconomic status, and some studies done in both Pennsylvania and New York, kids who grow up in a disadvantaged environment have poorer language skills and decision-making capacity and other effects on their ability to learn and remember. There's chaos in the home, a greater helplessness and distress as a result of being in a chaotic home environment, and as a result the prefrontal cortex and amygdala are not developing normal, and there is poor self-regulatory behavior, as well as obesity, elevated blood pressure, and cardiovascular reactivity, systemic inflammation, which is lasting at least into young adulthood and probably for the rest of their life, and poor dental health. And these are some of the people who are involved in these studies. And then finally, there's the classical, very important study of adverse childhood experiences by Robert Anda and Vincent Felitti. He's at the Center for Disease Control, and Vincent Felitti is in the Kaiser Permanente health system, middle-class health system in California. And